This video is going to offer an overview of some genetic and molecular topics such as DNA, RNA, protein, how this affects the proteins which the cells produce, which then affect our outward appearance and our function, our phenotype. Obviously, as these videos play in the background, teaching genetics, I have a lot more videos on this and other topics, and so feel free to consult those. So the idea is, why are we the way that we are? I mean, how is it that our cells, once again, cells are these microscopic structures, but nevertheless, they know how to make eye proteins and promote vision. They know how to form a brain in an embryo. How on earth could they know that? Because it's just 23 molecules, which we call chromosomes, which are present in the nucleus of the sperm, and 23 molecules, which we call chromosomes, which are in the nucleus of the ovum. This then has the plan for a person. Not only is this fascinating just from a biological standpoint, how could 46 molecules essentially program a person? But obviously, these cells, you know, in some embryos develop into fruit flies or into oak trees or into fungi. But in us, not only do they develop into humans, but humans with a mix of the features of our parents. So we inherit not only information on how to be human, but a specific, kind of human with traits from our individual parents, which makes us, individuals. So not only our skeletons and our hair and our blood types, but also our brain and aspects of our personality. So how is it that molecules can code for life? All right, well, this is certainly a profound question. Well, certainly there are specific instructions. We call these genes. And genes are coding for specific proteins, or perhaps RNA molecules, as I'll mention later. Well, how on earth could they do that? Well, as I'll go through in a second, the DNA, which sits in the nucleus, it has small molecules in essentially a sequence. And this sequence is so important that if you change the sequence, that will ultimately then lead to different proteins potentially and affect life, the risk of disease, etc. So in this video, I have a number of them, as we'll see it again at the end. The hemoglobin that could be made for red blood cells is either normal or abnormal, potentially resulting in sickle cell anemia. And the cause would be a change in the instructions in the nucleus, and this would be DNA instructions. That set of DNA instructions will then get copied into RNA, and if someone has a mutation, a change in the DNA, this will lead to a change in the RNA where it is not the normal RNA codon. And then when we build a protein following the instructions, the tiny molecules which make up the protein, the amino acids, will then be changed. So instead of the glutamic acid, which is the normal amino acid at a specific spot, then the mutation valine, the amino acid valine results instead. And so thus, if we want to focus on, say, the individual and their phenotype, the trait that they have, this is determined by cells. So sickle cell anemia, for example, would be determined by the abnormal shape of cells. If this is a normal red blood cell, and then this is an abnormal red blood cell. And then the abnormal function of the cells was determined by the abnormal protein. And the proteins were then constructed by an abnormal sequence of RNA molecules, an abnormal sequence of DNA molecules, and the DNA molecules form chromosomes in the nucleus of the cell. And so this is what I hope to explain in this video. How is it that DNA on chromosomes can code for RNA, which codes for protein, which then determines our phenotype, the function of our cells and our outward appearance. So just, some basic thoughts. First, this DNA is so important with all of its information, those genes that I had mentioned, including here the gene for for hemoglobin. It is so important that the topic of mitosis, which I'm sure you've studied in school, mitosis is the process by which we make sure that when a cell divides, each of the two daughter cells gets identical sets of DNA instructions. So in the nucleus of a cell, there's this DNA. Now, DNA is incredibly long. It's a very thin molecule. But in one of your cells, just one, there's almost 6 feet of DNA. You have enough DNA in your body to go to the moon and back several 100,000 times. So we have this incredible amount of instruction here. And when cells divide, the process of mitosis is how can we order this in such a way that we can be sure that each of the two daughter cells will get the exact same sets of DNA instructions. So if the DNA molecules are color-coded here, we want to make sure that each of the two daughter cells will get, say, a copy of the largest chromosome, chromosome 1, that you got from your father. Also a copy of chromosome 1 you got from your mother, a copy of chromosome 2 from both your father and mother. So mitosis is making sure that each of the two daughter cells then get identical sets of these DNA instructions. That's how important they are. And if we wanted to talk at a chromosomal level, changes then can have profound implications. So if we look at what is present in the nucleus of a zygote, once a sperm fuses with an ovum, there are 46 molecules, 23 of which came from the sperm, 23 of which came from the ovum. We call them chromosomes, and chromosomes are molecules of DNA. The DNA is very long and thin, but it's condensed around a protein scaffolding to form these chromosomes. Now, this is so important that if you had an extra chromosome or were missing one, almost always that results in death, unless we're talking about the sex chromosomes. There are a few exceptions, like having a third set of chromosome 21. But these instructions are important. So changes such as, missing a chromosome or having too many, typically lethal. Also potentially serious are changes to the structure of a chromosome. So if you have 46 chromosomes, but one of them is broken at the tip and you're missing part of it, or if one chromosome gets an extra little piece, or if the part of one chromosome then goes to another chromosome, because this is the instruction book for you, this can have devastating consequences in health, can result in cancer, can result in birth defects, can result in infertility for a number of reasons, which I don't mean to go in here, but one of which is simply, not only do we need to make the right proteins, we need to make the right amount. So notice that this cell, let's say it has the normal dosage of genes. It has 2 chromosomes, like it's supposed to. It has this much gene product for a specific gene, say the hemoglobin gene. If you were missing a chromosome, you would only have half the normal amount. If you had an extra chromosome, you would have triple the amount. So it turns out that not only making the right proteins is important, but then also making the right amount. How is it that our cells can do this? Okay, well, let's step backward and consider the DNA in the nucleus. It is in the nucleus of the cell that the DNA is housed. And I'm just going to focus on that. There is actually a little bit of DNA in the mitochondria as well. And actually, that can contribute to inheritance. But let's focus on the overwhelming majority of the DNA, which is in the nucleus. This is wrapped in then the nuclear membrane. So this is important. And we have to send messages into the nucleus to change which genes we're using at any one time. So for example, estrogen and testosterone bind to their receptors, actually go through these holes, these pores in the nuclear membrane. They bind to the DNA and then turn on genes, change which genes are active. And so that nucleus is kind of the command center of the cell, and we want to regulate which genes are on or off at any one time. Now, this DNA, this is a what's called a polymer. A polymer is when a long molecule is made by repeating subunits, which we refer to as monomers. So DNA, as I said, is incredibly long. The DNA in just one cell in your body would be almost as tall as you are if you could stretch it out. And as you can see, each of these rectangles represents a monomer known as a nucleotide. Excuse me. And so we get our DNA molecule by attaching one to another, to another, to another, with more than 3 billion nucleotides representing the genome of humans. And so once again, this huge DNA genome is made of chromosomes, and chromosomes are made of individual molecules of DNA. These DNA molecules are very long, but as then I said, they are what are called polymers made of repeating monomers. And if we were to study then cell division, one of the important phases which has to happen prior to cell division is if one cell is going to divide and make two, that if this represents the DNA molecule, which is, say, the chromosome 1 you got from your mother, well, before this cell can divide, this DNA molecule has to replicate, because each of the two daughter cells will need a chromosome 1 you got from your mother. These genes and the right number of genes, these are critical. And so what's called the S phase prior to cell division is when we take our DNA molecules, which are normally in the structure of a double helix, we unzip the double helix, and then we add the new monomers, the new nucleotides, so that by the time the process is done, where there was once a single chromosome 1 that you got from your mother, now there are 2 chromosome 1s that you inherited from your mother. And when this one cell divides to make 2, then each of the 2 daughter cells can get 1. And so in the process of mitosis, we copy all of our DNA to make 2 identical sets. And that way, if you were to go into, say, a cheek cell in your body, cheek cells have the same DNA as your muscle cells and as your brain cells, because every time a human cell replicates, then each of the 2 daughter cells get identical copies. Another thing, we don't have to go to it in enormous detail here, but one of the ways that this is regulated is there are 4 of these nucleotides, and they attach to each other across the double helix in a specific way, so that the adenine only binds to thymine, the thymine only binds to adenine, the guanine only binds to cytosine, and the cytosine only binds to guanine. This is what's known as complementary base pairing. And as a result, whatever the order of the DNA bases in one chromosome on one strand, this would then complement the DNA bases on the opposing strand. Well, that's DNA. But we then, from the DNA, we have to make copies of the DNA, and these copies are in a related molecule called RNA. RNA nucleotides are just a little bit different. So here we have an oxygen in this sugar known as ribose, but notice there's no blue dot here. So this is what's called deoxyribose. It lacks the oxygen attached to this carbon. And so, in addition to DNA, a nucleic acid, we can make a related one, RNA, which is just a little bit different. The process of making RNA copies of the DNA is going to involve unzipping the double helix of DNA, indicated here in red, and then making this single-stranded RNA copy of it. The main thing to be achieved here is that the DNA sequence has a start code, all right? And so when we look at the DNA sequence, the adenines, thymines, guanines, cytosines, somewhere in here there is meaning. And one of the possible meanings is to have specific sequences which say a gene starts here. And that type of thing we call a promoter. So when your cellular mechanisms come across promoters, which essentially say a gene starts here, we can start the process of transcription. And in transcription, we unzip the DNA and make an RNA photocopy of one of the strands. It is the RNA which will then leave the nucleus and go out into the cytoplasm where the protein will be made. So here in the process of transcription, RNA nucleotides are interacting with the DNA nucleotides. The way that one could tell is that the sugars are different. So here in red, I'm indicating the deoxyribonuclease, I'm sorry, the deoxyribose of the DNA strand, and in green, the ribose of the RNA strand. So these are different sugars, and RNA, thus that ribonucleic acid, named after ribose, is different from DNA, the deoxyribonucleic acid here. But the sequence of the RNA bases is determined by the sequence of the DNA bases. So notice that this pink adenine is binding to this blue thymine. And then here, the same thing, that pink adenine is binding to thymine. And so that this is not a random sequence. It is the sequence of the DNA nucleotides, which is determining the sequence of the RNA nucleotides. This white base here is because while DNA has a nucleotide for thymine, in RNA, thymine is not an RNA nucleotide. It uses uracil instead, a different one. And so in the process of transcription, an RNA copy of DNA molecules are made. Now, once this RNA copy is made, the benefit is it's short, while a DNA chromosome is very, very long. I mean, the chromosome 1, if you were to stretch it out, it'd be about that long, just a chromosome 1. That's huge. RNA molecules are smaller copies of genes that can then leave the cell. Now, we understand RNA so much better than we did, like even when I was in school. One of the things that we understand is RNA is so complex. It's capable of these complex ways of folding. And while it used to be thought that essentially, the DNA, which was meaningful, coded for RNA, which coded for protein, as I will get to, we now understand that a lot of our DNA codes for RNA, but not protein. RNA is quite a complex molecule. And actually, RNA molecules do a lot of tasks inside the cells. So while one of the roles of RNA is to be an intermediary between DNA and protein, very often RNA is its own end product. Now that's a little bit more complex, but just wanted you to hear that. So the RNA then leaves the nucleus of the cell and goes out to structures known as ribosomes, where proteins will be made. How is this possible? Well, the only thing you need to do to make a protein is to know which amino acids you put in the right order. So there are 20 amino acids to choose from, all right? And so if you're going to make a protein, which of the 20 do you start with? Which of the 20 is the next one? Which of the 20, comes next, etc. And that's what the RNA code, which is a copy of the DNA code, that's what it does. All right, the RNA code then tells the cell which of the 20 amino acids is appropriate. Now, it's a triplet code Because if you think of codons, which are, meaningful packets of RNA nucleotides, codons are what are called triplet. There is the first nucleotide, the second nucleotide, and the third nucleotide in the RNA codon. Once you have this triplet, there is meaning. So any of these triplets, they mean something. They mean cell, put leucine at the next spot in the protein. These words here, and a codon is essentially an RNA word, they tell the cell, put a proline here, put an arginine here. So remember that sickle cell gene had made a mistake, because what should have been aspartic acid being put in one place instead was valine. All right, and so if you, instead of sending instructions to put this, it was glutamic acid. My fault. So instead of putting glutamic acid in one spot, if you put valine, that's a mutation. That's a different nucleotide. Different amino acid sequence. And it matters because amino acids, they vary. Some are non-polar and don't interact with water. Some are polar, some are acidic, some are basic, some have aromatic rings attached. So which amino acids you put in which order in a chain, it makes a difference. A mistake, a mutation could, actually be so serious that this would end up having, negative health consequences, or perhaps even causing death. So for example, there's a very important protein known as P53. More than any other protein, it helps prevent us from getting cancer. And if you said, ooh, I definitely want to make a protein which prevents getting cancer, how would I make it? Well, the answer is you have to put a phenylalanine next to a phenylalanine, next to a serine, next to an asparagine, next to leucines, next to these amino acids in this order. So once again, there are 20 amino acids. I've colored them with different pieces of paper because once again, some are polar, some are non-polar, some are acidic, or some are basic. So it matters which of these you put in. Now actually, this one here, this is the code for the protein which codes for the chloride channel, which when mutated would result in cystic fibrosis, all right? And so cystic fibrosis, there was that one spot which is missing in the majority of people who have cystic fibrosis. And so this is an important sequence of amino acids. Not having the right amino acids could result in this disease. In the same way, if this then is the P53 code, and it should have a glutamic acid, then a glutamic acid, a threonine, an arginine, etc. The red piece of tape with letters, those are the letters of the incorrect amino acids, which some individuals had. So some individuals had a proline in this spot instead of a histidine. But each of these red spots came from a patient who had cancer. So putting the wrong amino acid in this chain, it could actually have life-threatening consequences. So then what's sometimes called the central dogma in biology is that chromosomes like this one, they have genes made of DNA. The DNA is then transcribed into RNA. And then the RNA, let's imagine the RNA, the mRNA is this blue molecule. It is then translated to make a protein. This RNA code, and once again here on the blue molecule, is instructions, those triplet codons which say, hey, put this amino acid first, put this amino acid second, put this amino acid third. Those codes are read at a structure called a ribosome. So ribosomes are very important parts of the cell. This is where proteins are made. This is where the code in the nucleotide order of the mRNA is converted into an amino acid order of the protein. The RNA says, hey, bring this amino acid first, and so that amino acid is brought first. The RNA codon then instructs the cell, bring this nucleotide here second. That nucleotide is brought there second. The two nucleotides are stuck together, all right, starting to form the beginnings of a chain, all right? And then the process will repeat itself over and over until a protein is made, where more and more amino acids are added, but not in a random fashion. Amino acids are added in the precise order that the DNA nucleotides prescribed, and then the DNA nucleotide order was copied into an RNA nucleotide order. So those processes that I just described, they will continue until you get to a codon of the mRNA, which doesn't have a corresponding amino acid. It's what's known as a stop codon. In that instance, then the protein chain will now be completed. No more amino acids will be added, and now this chain of amino acids is the protein whose amino acid order was exactly what was prescribed by the DNA. So this is important. I need to know how to make keratin for my hair, or opsins for my eyes, or collagen for my connective tissue, or hemoglobin for my blood, or proteins which stop cancer like P53 or BRCA. And if this is the protein with the correct amino acids in the correct order, it was the DNA which had the instructions on that order. And the DNA then is passed down to each cell, to each generation. The DNA can then be copied into RNA, and then the RNA at a structure known as a ribosome is now translated into an amino acid sequence, which makes up then the proteins. If you changed this code so that this protein was not made, well, that change in the DNA sequence would be called a mutation. Now, there are lots of effects of mutations. Some mutations, well, really don't have much of effect. So, for example, if you notice GUU is a word that means valine, GUG is a word that means valine. So if you had a mutation changing GUU to GUG, then the result would still be to insert valine at that spot. So that mutation specifically didn't have a major impact on the cell. But you can have some mutations now change the amino acid which is inserted in the protein from, say, leucine to another one like proline. Or maybe it would be converted to a stop codon, meaning that the protein would be shorter than it otherwise should be. There are a number of possible effects of mutations, but one of the effects that they can have is to change the amino acids in the protein. And as I've mentioned, that type of mutation can have serious effects for living things. All right, and so just as I start to wrap up, so if you want to not have cancer, you need to make the tumor suppressor protein P53. But in half of all human cancers, this protein is mutated. And so therefore, this is clearly important. So we have chromosomes in the nuclei of our cells with the P53 genes, with the DNA instructions on how to make them. And every time our cells divide, we copy these P53 genes and pass them on to then the daughter cells. These genes have a specific nucleotide order in the DNA molecules. We don't want the nucleotide order to be changed. A change or a mutation means that if the DNA sequence is changed, that this could change then the RNA sequence which is copied from it. So the RNA codons which leave the nucleus and go out to the cytoplasm of the cell could be different than what was originally intended. When the RNA codon is then translated into an order of amino acids, then in the mutated cell, there could be a different order of amino acids, say proline the normal amino acid might be replaced by threonine. And this could have a negative impact on the cell. So for example, a mutant protein might not function, or it might not function the same. And then this could have a negative impact on the cell. And so this might result on this person having cancer. They have cancer because a mutation on the chromosome in the p53 gene meant that there was a different DNA code, which led to a different RNA code, which led to a different amino acid code in the protein, which then meant that the cell wasn't making the normal protein, which could then have various effects. Now, we have lots of genes, over 20,000 of them. And so not only do we have to correctly make the p53 protein, or else we'll develop cancer, we also then have to make the correct BRCA1 protein, or else we can develop a cancer. A number of cancers are possible, with breast cancer being a common one. And so this was kind of an overview of what's called the central dogma of biology, where it is DNA, which is the code for living things that gets replicated with each cell division. But then DNA can code for RNA and transcription, which can then be translated into the amino acid of a protein, and then proteins can then perform the various functions in cells. And as such, then this is a good introduction into some central tenets of molecular biology. Obviously, there's far more. Let me just mention two interesting ones. First off, it's not just a question of genes and having the right amino acid order. We have to be able to regulate the genes, turn on genes at the right moment. And so the regulation of genes and the proteins which stick to DNA, called transcription factors, which help to regulate genes. This is certainly a major area to study in molecular biology. Then we're continually learning new things. So for example, while mutations are, say, permanent changes to the nucleotide, where even if the DNA nucleotide order is the same, nevertheless, we could have some changes of the DNA, which could be influenced then by our environment, which, while it wouldn't change the order of the amino acids, it could change, however, the gene's activity. And so this is something which isn't, inherited in the sense how DNA is packaged, how densely it is controlled. This can affect the expression of genes, and thus our appearance and our traits, etc. So this was just kind of a quick overview of some central tenets in how our cells and genome works at a molecular level. Obviously, I have a number of videos which go into this in greater detail, if that is an interest.